Thank you so much for that introduction, Ricardo, and also for the invitation to be here today. I'll be presenting preliminary outcomes from a project funded by the Flexible Funds program at Leibniz University Hannover. And the goal of this project is to develop a process for publishing biodiversity knowledge in a format that is readable and usable not only by people, but also by machines. And later in my talk, I'll be explaining why this is something we should all be motivated to do. The project is a really cool collaboration between biodiversity researchers and computer scientists. And as somebody with interdisciplinary training as a systems thinker, I'm serving as a bridge between these two disciplines, though I don't have formal training as a computer scientist. So this has been a really fun and exciting challenge for me. But fortunately for me, I'm working with a really awesome team of people who are equally enthusiastic about this interdisciplinary initiative. So I want to take a moment to introduce you to them. So as I said, this work is really the result of a very collaborative process between biodiversity researchers at JLU and LUHA, and computer scientists from LUHA and the Leibniz Information Center for Science and Technology, and the Open Research Knowledge Graph Initiative, ORKG, which I'll be explaining in more detail later in my talk. So to begin, here's just a short outline of my talk to orient you to the different material that I'll be discussing today. So I'll start by giving you some background information on the importance of biodiversity synthesis research as a tool that can help us gain insights into the complexities of biodiversity data. And then I'll also talk about some of the challenges that we as researchers experience when we try to conduct synthesis research. Then I'll introduce you to the Open Research Knowledge Graph, or ORKG, which is a hybrid human and artificial intelligence-driven platform that's beginning to address some of the challenges of scholarly communication by making information readable by people, but also by machines. Then I'll explain some of the motivations behind creating machine-readable scientific information and explain how we're using a published agroecological study by Ricardo and colleagues as a case study for doing this. And then I'll close by just briefly sharing our vision of how this approach could and should be applied more broadly in the field of ecology and describe some of the next steps that we're taking in that direction. So let's start by talking about the importance of synthesis research, particularly in the context of biodiversity loss. And then we'll look at some of the challenges that we face when trying to do systematic reviews or meta-analyses or other types of synthesis research. So developing actionable insights into biodiversity research is extremely important in the context of biodiversity loss. We're losing species at an unprecedented rate, and this trend of biodiversity loss caused by human pressures has major implications not only for the environment, but also for us as humans. So this figure, based on a recent paper by Leclerc and colleagues, illustrates the dramatic loss in terrestrial biodiversity that we will witness unless we take ambitious and focused conservation action. So continuing with business as usual is not an option if we want to avoid catastrophic biodiversity loss. But there's also a hopeful message here that if we enact and sustain very strong conservation measures now, we can slow and potentially even reverse this trend of biodiversity loss. Currently, one of the main challenges to developing these types of conservation strategies is disentangling and making sense of the very complex and multifaceted nature of biodiversity data. So with hundreds of possible factors that are driving these observed patterns across space and time, it's really a challenge to identify what are the main drivers that steer ecological communities, much less to compellingly convey this information to policymakers and the public. So for example, when we think about biological conservation or biodiversity conservation measures, it's not uncommon that we have to address multiple factors simultaneously, like climate change or land use changes or pollution. And so it's not always clear how to deal with these different factors and their interactions. And even when we can obtain meaningful insights, the existing tools and methods that we have struggle to generalize patterns from this highly correlative and context-dependent nature of this data. So this is where synthesis comes in. So synthesis research that integrates findings from multiple biodiversity studies can help us to gain insights into this complexity, by identifying the patterns and the processes that underline the trend of biodiversity loss. And it's through elucidating these patterns that we can start to predict some of the likely scenarios of biodiversity change, and also to inform policy recommendations for biodiversity conservation. Clarifying these patterns through synthesis research can help us to succinctly message the benefits of biodiversity in a way that resonates with policymakers and the public. And this recent global synthesis did exactly that. By synthesizing data from hundreds of sampling locations around the world, this publication delivers a clear message about the importance of biodiversity for crop production. But despite the benefits of syntheses like these, they're increasingly challenging for us to conduct. So one major obstacle is the growing number of new biodiversity publications, which makes it really hard to identify relevant literature, and it diminishes the relevance of these syntheses over time. So we have decades of research looking at how biodiversity responds to different stressors, to different management practices, and that's great, but all this data is really hard, makes it hard for researchers to stay up to date on the latest findings and to identify clear trends and patterns that have broad scale applicability. And this publication flood only continues to increase in magnitude. So in 2010, there were roughly 200 scientific articles published annually on the topic of biodiversity loss alone. By 2020, this number increased to about 1,000 publications a year. So this roughly 5-fold increase in the number of scientific articles published each year not only makes it hard for researchers to stay up to date in their field, it also renders the relevance of syntheses fleeting in the wake of these new findings. So another way to think about this is if we take ourselves back to 2010 and we imagine that we're young, energized scientists ready to save the world, if we ambitiously read a paper every couple of days, we could capture all of these 200 publications. But fast forward to today, we would have to read at least 2 to 3 papers every day, including on the weekends, to stay up to date just in the field of biodiversity loss. And that's assuming that we're lucky enough to even be able to find all of these papers. And our antiquated method of sharing information doesn't do anything to help this situation. So fundamentally, the way that we present information really has not changed over the last several hundred years. So back in the 1600s, researchers were publishing information in a physical document format that maybe was a little difficult as a human to read. And so we've made some advances today. We have these visually pleasing PDF documents that are electronic, so we can easily share them. They probably have beautiful figures and illustrations. So as a human, it's easy for me to read, but it's still a document-based method of communicating information. And the other similarity between the way that we communicate today and the way that we communicated several hundred years ago is that these documents remain out of reach for machines. And that means that we can't take advantage of machine-based tools like intelligent searches or data filter functions that would help us to efficiently extract data from these documents. And this leads us to our second hurdle, which is the challenge of machine readability. So I've mentioned this concept of machine readability a few times, and what I mean is that you have this nice PDF that's easy to share, but most of the data in it would have to be manually extracted by a person if you wanted to reuse it. So while we have a number of great reference managers like Zotero and others, where you can upload a PDF to the computer, and the computer reads, extracts, and organizes the metadata of the paper, such as the title or the author list or the publication, into this nicely structured format, most of the results remain out of reach for machines. So you can't feed a PDF into a machine and expect it to process and structure the vast majority of information. And this information, that's because this information is not machine readable. And this poses significant challenges to synthesis research because it puts the burden of data collection and organization on the human researcher. So for instance, if I wanted to conduct a meta-analysis and I wanted to include the results presented in this figure, it would be pretty hard for me to do that because there are no quantitative values that I can just transfer over to an Excel or CSV file. I would have to use a plot digitizer or some other data extraction tool to estimate the values from this figure, and then compile it into a data frame where I've collected information in a similar way from other publications. In other instances, I might need to interpret and manually collect additional information that's presented in unstructured text format, keeping in mind that this format probably varies dramatically across papers. And a final challenge is that the traditional practice of publishing as a PDF makes it almost impossible to update syntheses or any other article once they're published. So you've put all of this time and effort into publishing this incredible new synthesis that has a great message, but as research advances, which we have seen occurs at an alarming rate, we now have a publication that's becoming outdated. So now a novel synthesis has to be conducted to capture the latest findings, and this results in yet another publication and data set for the community to keep track of. So to summarize, our current approach of communicating scientific information in these pseudo-digitalized PDF formats presents several problems. One, it contributes to a publication flood that makes it difficult to keep track of all of the new knowledge and to figure out what information should be included in a synthesis. Second, the data is not machine readable, so it has to be manually extracted, which is a laborious process. It also results in poorly structured articles that are not tied together by consistent or clear language or structure. And finally, we end up with a static PDF article that can't easily be updated once it's published. So in the project that I'm going to describe to you today, we're going to be focusing on these first three challenges. And they really have a big impact on the amount of time that it takes to conduct synthesis research. So here is a study that actually looked at the time investment of conducting a systematic review. And they found that the number of hours it takes to conduct a review is highly related to the number of citations that you retrieve in your initial literature search before you apply any exclusion criterion. And on average, they found it takes about 6 to 12 months to complete a systematic review. And that's assuming that this is your full-time focus. But as we know, most reviews are undertaken on a part-time basis. Nobody has 40 hours a week consistently to dedicate just to this process. And while these data are based on the amount of time it takes to conduct systematic reviews in the medical field, they provide a good proxy for the amount of time it could take to conduct a biodiversity synthesis. So if we remember that we're publishing about 1,000 publications a year just on biodiversity loss, you can imagine that by the time you're ready to submit your review for submission to a publisher, you're already missing relevant information. And of the total time that it takes to get a systematic review from conception to submission, about half of that time is devoted to pre-analysis literature search, retrieval, and database development. So you can imagine if we could create publications from the outset that are machine readable, we could leverage the power of machines to drastically streamline the process of data collection and organization and reduce the amount of time that it takes to conduct a meta-analysis or other synthesis. So the takeaway is that we have a lot of really complicated data. It's spread out over thousands of papers, and we don't quite know what to do with it. And this results in a lot of inefficiencies when it comes to conducting synthesis research. Well, this kind of sounds like a problem for a machine, right? So we've increasingly seen how computers and artificial intelligence initiatives are helping to address complex societal challenges. And the world of academic research is no exception. So maybe you're familiar with this visualization tool called Connected Papers, which provides an overview of your field and helps people to stay up to date with the latest work. So this can be a really great tool for navigating the publication flood. Another really interesting initiative is High Knowledge, which produces interactive networks of hypotheses in the field of invasion biology. So think disturbance hypothesis, for example. Using this tool, you can see the number of publications that support, question, or remain undecided about a specific hypothesis. So this tool provides strong insight into the state of a specific domain of research, but its specialization limits its broad applicability and reuse. So we'll give it a light green check mark here because it does help navigate the publication flood, but only for certain fields of science. Stencilla is another really interesting tool that provides an office suite that's designed specifically for researchers. So it allows you to author these interactive, data-driven publications in visual interfaces that are similar to those in a conventional office suite. But it's designed specifically for reproducibility. And it's probably the state of the art in this aspect because it makes pretty much any quantitative value and associated statement fully reproducible by providing the data and the code that underlies that information. However, the data in Stencilla is not machine readable, and while it brings structure to articles in the form of statistical code, it does not ensure consistent use of language across papers, so again, we'll give it a light green check mark in this column. So you can clearly see that there is a gap when it comes to addressing some of the obstacles for conducting synthesis research, particularly related to machine readability. And there are efforts that are making data machine readable, but they tend to be focused mostly on bibliographic metadata, or they're very discipline specific. So there's a need for tools that promote machine readability across disciplines. And so here's where the Open Research Knowledge Graph, or the ORKG, comes in. It's a recent open access initiative that brings the elements of machine readability and language clarity to scholarly communication. And it does this by fully digitalizing and semantically structuring scholarly information. So by fully digitalizing scholarly knowledge in a structured format, this online hybrid human and artificial intelligence platform makes information accessible not only to people, but also to machines. So that means that a computer algorithm can now process and structure results like those presented in this figure. And once machines can read and process this information, they can help us interpret and compare scientific findings and distill patterns from complex and heterogeneous information, which sounds a lot like biodiversity data, right? Making this data machine readable also makes it easier for people and for machines to access and reuse this information across disciplines, which further facilitates synthesis research. And the way the ORKG does this is by using this knowledge graph format. It structures knowledge and information in the form of semantic knowledge graphs. And these are just semantic or language-based networks that use formalized language to illustrate how different objects or entities or concepts are related to one another. So here's a very simple example where we're using a knowledge graph to illustrate how a document, which happens to be an article, has different kinds of information. So it has information about an author that relates to a particular person. It has publication date information that maps to a specific time or date, and it has an image that maps to a particular object. And by relating information in a knowledge graph format, we're structuring and defining it in a way that can be processed by machines. And knowledge graphs produced in the ORKG can address much more complicated information than the simple bibliographic information that I just shared with you in the previous example. And we can also view knowledge graphs from the ORKG in a tabular format. And we call this a comparison. So for example, we can address a question related to biodiversity or agroecology. For instance, how do legume intercrops affect grain yield? And then if we look at these different columns, these represent different studies that address this question. And now we have different rows of information that have different information or characteristics about these studies. So as a human reader, I can look and quickly see, okay, they're all addressing the same research question here. They're looking at how legume intercrops affect yield. And I can look at similarities and differences across cropping systems, study location, experimental design. We can even provide a qualitative summary of how the treatment performed overall. So did legume intercrops tend to have a positive, negative, or neutral effect on yield? And what's really neat is that not only can I easily read this as a person, but all of this data is also machine readable. And we can also look at the quantitative values associated with the yield of, in purple here, I have highlighted the yield from the treatments with the legumes, and we can compare that to the data presented in this yellow box, which maps to the control, which were grain monocultures. And we can also look at this information in the form of a figure. So here on the y-axis, we have grain yield, and on the x-axis, we have each of the different studies that were included in that tabular comparison that I just showed you. So the blue bars show us the yield associated with the control, which again are grain monocultures. The red bars show us the yield that came from the legume intercrops. And when we see a star, that denotes a statistically significant difference between the control and the treatment. And so, as you can see, in most cases, there's no significant difference between the grain yields and the monocultures compared to the intercrops, which suggests that enhancing biodiversity by growing legume intercrops does not come at a cost to yields. However, in 3 instances, we do see that legume intercrops had significantly lower yield than the control, which suggests that more work needs to be done to optimize these systems so we can exploit the other benefits associated with legumes, such as soil fertility. And this visualization is a great example of the power of the ORKG platform because it allows us to see trends across studies, and it gives us an overview of the state of the art of our field. So for example, in this visualization, we can see that on average, intercrops perform on par with monocultures, but there are a few instances where they underperform. So now we could look at these specific instances and start asking why they may have underperformed in these situations, and or identify similarities in the studies where they underperformed. So for example, were they conducted in a particular location? Did they use a particular legume crop species? And this knowledge would shed light on the conditions needed for intercrops to provide yield benefits, and could also help us develop more targeted research questions focused on improving the performance of these systems. So this is just a simple and specific example, but you can imagine how we could use this approach in the ORKG to address other biodiversity topics. So for example, how does a particular species respond to climate change? Or what are the impacts of a biodiversity conservation strategy across locations? So it's a useful approach for us to get a general and quick sense of how these systems are performing. But despite its potential, the ORKG has not been broadly explored or leveraged in the field of ecology, or more specifically, in the subfield of biodiversity research. And a key barrier to scaling its use is getting data and information from these scholarly publications into the platform. And remember, most scientific data currently is published in a PDF format that is not machine readable, so it can't automatically be fed into the ORKG. Rather, the data has to be manually extracted and entered into the system by scientific experts, by people. So one way to address this issue would be to create a process where researchers can publish their data in a machine-readable format from the outset, so that new research findings could be automatically captured by the ORKG every time a paper is published. So this would remove the middleman, so to speak, and circumvent the need to go back and manually add the information into the platform. And in addition, by allowing authors to formally describe their own work, this approach would also avoid curator bias that can occur when other people are trying to go back and interpret your work for you. So this brings us to our current project, where we're creating tools that would allow researchers to do exactly this: publish their data in a machine-readable format from the outset. And an equally important goal of our project is to convince ecologists of the benefits of doing this. So I hope by the end of the talk, I'm able to persuade all of you. So now I'm going to walk you through the process that we're developing to make the research data and information machine readable. And I'll do this by presenting a case study that uses an already published paper in Ecological Applications written by Ricardo and colleagues. And this paper looks at the effect of landscape composition on the incidence and abundance of different pest species. So I'm going to show you how we can transform a research article from a PDF that's unintelligible to machines into a highly structured, machine-readable format that can be automatically harvested by the ORKG. And once this information is in the platform, this data is ready to be directly downloaded and reused. So you wouldn't have to manually harvest this information. So today I'm going to take you behind the scenes of this process to show you how we make this transformation to machine-readable literature. But before I do that, I want to emphasize that from your perspective as a researcher who may want to implement this process, this will ultimately only require adding a few lines of code to the R or Python script that you use to create the figures and the statistical analyses in your research article. So this process does not add to the already intensive publication process. It really just requires a small amount of code, which I'll demonstrate in a few minutes. But for now, I'm going to take you behind the scenes of how we do this by focusing on a single research contribution from Ricardo's paper, just as an example. But we're going to apply this method to other figures and information from Ricardo's paper presented in the text, so that at the end, as much of his article is machine readable as possible. So let me show you how it's done. So first, we started by identifying the information that we wanted to ingest into the ORKG platform, and we'll refer to this information as our research contribution. And in this example, we focused on this particular figure that shows the relationship between the proportion of meadows within a 250 meter radius of an experimental field plot and the incidence of aphid pests in that plot. And we also wanted to capture the outputs of the statistical analyses that were used to describe the trends presented in this figure. And that's the data I'm really going to focus on showing you in the next parts of this example. So this brings us to the second step, which is to fully describe the processes that underlie the information that we want to capture. So in many cases, the research contribution that we want to ingest into the ORKG is the result of multiple processes. And in this example, that was indeed the case. So there were several different processes, but that were related to one another that we wanted to capture. So in addition to describing the output figure, which I just showed you, we also wanted to capture the R script that Ricardo used to create the figure and also to run his analyses. And this included running a linear mixed model. And so in addition to capturing the quantitative output from the model, we also wanted to describe this process of model fitting. There are also summary statistics that were run on the model that we want to capture, in addition to an ANOVA and model predictions. So by accurately describing and capturing the quantitative values of these different analyses in a machine-readable format, we're going to make it much easier for another researcher to come along and reuse this information in a secondary analysis. And one way that we can bring structure to these processes and the relationships in the ORKG is to apply what we call templates. And this was the next step in describing Ricardo's paper, was to develop some templates to bring some structure to these different contributions. And you can think of templates as a kind of fill-in-the-blank form. So here is an abridged example of a template that we use to describe the linear mixed model fitting process in Ricardo's paper. So we wanted to capture and structure information about the input data that went into the model, so the raw data that he collected in the field. We also wanted to structure and describe the model output data. There was significance testing that produced quantitative values that we wanted to capture. And we also wanted to describe the process of model fitting and really make explicit the different components of this linear mixed model. And then, of course, there was also an output figure to be captured as well. So as you saw in the previous slide, the relationships of these different processes can quickly become quite complicated. So one of the goals of the templates is to make it easier for subsequent ORKG users to add their research contributions to the ORKG without always having to map out these relationships. So for example, if you wanted to come and add your information about a linear mixed model that you conducted on another data set, it would be as simple as calling this template in your code, in your R or Python code, and then the data would automatically be mapped to this template structure. And this is important because if independent researchers describe their methods in a similar way, we can ensure structure across publications. And this addresses some of the issues that I mentioned earlier with inconsistent use of language and lack of structure across current publications. And one of our goals in this project is to develop templates that describe other analyses that are commonly used in ecology and biodiversity research. So for example, ANOVAs or t-tests or path models, so that as much of this data can be easily described and structured as possible. So the next step in transforming Ricardo's paper into a machine-readable format was to structure the description of his research contributions in a semantically or language-based way. And we did this by integrating ontologies. And we could have an entire seminar just on ontologies, but for our purpose, it's enough to understand that these are formal vocabularies that are used to define concepts and describe the relationships between these concepts. So you can kind of think of them as like a taxonomy for words. And here's an example of an ontology for the word aphids. So at the broadest level, we're describing something that's a thing, it's an entity that belongs to these different subgroups. And you can even see part of the actual taxonomy for aphids. And each of these words and phrases that are included in the aphid ontology have their own ontologies. So we can really start to specifically describe and connect different concepts to one another. And the best way to understand why these ontologies are an important part of the machine readability process is with an example. So let's say that I'm a researcher that wants to conduct a synthesis on how landscapes affect aphid populations. And I want to include information from Ricardo's paper, where he described the relationship between landscape composition and aphid incidence. But I also want to include studies that report the effect of landscapes on aphid abundance or aphid density, because these terms also describe aphid populations. For me as a human, this is no problem. I can look at these phrases and understand that they're related concepts. But for a machine, these will appear as distinct and unrelated concepts unless I relate them in some way. So to a machine, aphid incidence is completely different from aphid abundance, is completely different from aphid density. At this point, the machine doesn't understand that each of these terms has a common denominator, which is aphid. So we need to make this information explicit to the machine. And this is where the ontology comes in. So if we map these terms to an ontology for aphids, it becomes clear to a machine that all of these concepts somehow relate to aphids. So now if I go into the ORKG and I say, hey, ORKG, give me all the information you have on aphids, I'll recover all of this data. So this is a very simplified explanation, but it demonstrates how the use of ontologies in the ORKG can resolve some of the issues that arise from machine readability when we use different language across publications. And that's because it allows us to connect or group terms that are seemingly distinct to computers. So while we as humans may know that we're referring to related things or concepts, we need to make this information explicit to machines if we want to more fully leverage them in synthesis research. So now the next step is to write the R code that will produce these machine-readable descriptions of the analyses and the figures described in the PDF publication so that they can be automatically ingested into the ORKG. And this is the part where you, as the researcher who wants to publish your article in a machine-readable format, comes in. So what I'm going to describe next are some of the examples of the R code that you would add to your existing script to create machine-readable descriptions of your data. And as I mentioned before, you'll see how this process really only requires adding a few lines of code to the existing script that you already produced to do the analyses and the figures in your paper. So for example, here we have the R code that Ricardo originally wrote to create the linear mixed model that was used to describe the trends in this figure that we saw previously. And now we add a few lines of code showed in this box that will ingest the description of this linear mixed model into the ORKG in a structured and machine-readable format. So all this code does is call an existing template to describe a linear mixed model, similar to the template I showed you before. And then it's going to map information from Ricardo's code related to the model response variable, the fixed effects, and the random effects terms to this template. And this template will provide also a field for a label that's human readable. And by mapping the data to this template, we ensure that it's harvested by the ORKG in a structured format. And again, this template would be available for reuse by any subsequent researcher who also wants to describe their data in a similar way. And so here's what this structured format actually looks like in the ORKG. So we would have our human readable label here. So as a user, I can quickly get a sense of what the model is looking at. But then we also have our information about our response variables, our fixed effects terms, and our random effects. And these descriptions in red here are already in machine readable format. So if there was another model from another publication that talked about any of these terms, the ORKG could easily link them and send me all of that related data. And the way we do this is by applying those ontologies that I mentioned earlier. So for example, if we look at this phrase, aphid incidence in an agricultural experimental plot, we can break down the different phrases and link each of them to a separate ontology. So for example, aphids links to that ontology that I showed you earlier. We can also link to an ontology on incidence. So now the computer understands that we're talking about the incidence of this particular pest. And we also show how these different terms are related to one another. So in a way, we're kind of acting like a language translator. So we're translating human language into computer language. So let me give you another quick example with quantitative data to give you a sense of how this could be really useful for conducting a synthesis where you're trying to compile lots of quantitative data from different publications. So here's the original code that Ricardo ran to summarize the output of the linear mix model. Now we just add a little bit of additional code to produce a machine-readable description of the quantitative summary output, which looks like this in the ORKG. So now think if you were to see this information in a traditional PDF publication, it would be presented probably as a table, but in text format. So if you wanted to use any of the information provided in this table, you would, as a person, have to manually extract this and compile it into a data frame. But here in the ORKG, it's already in machine readable format. So it's ready to be downloaded and reused in a secondary analysis. You can see here there's this feature called export as CSV that allows us to automatically download this data. There's another function in the ORKG that would allow you to directly send this information as a data frame to R. So you can right away start doing your secondary analysis and collect information like this from other publications as well. So the final step in this machine readable transformation process is linking the data that we produced in these R codes to the manuscript DOI. And this allows the information to be automatically captured by the ORKG using a simple DOI lookup function, which eliminates the need for manual data entry. And this is similar to what a reference manager, like Zotero, does for the manuscript's metadata. So you upload your PDF to your computer, your reference manager captures some metadata that's associated with the manuscript's DOI, and it structures the data in this nice format. And we all know the benefits of reference managers that make our lives so much easier when we're trying to compile our citations. But with the ORKG, what we're doing is we're creating a system that does this, but in addition to the metadata, this DOI lookup function also captures the results of the paper, the actual substance of the paper that we would want to reuse in a synthesis. And this is done simply by saving the data that we produced in these short R scripts in a standardized file format called a JSON file. And this is just a type of file that the ORKG can easily read. So then what would happen, so you write your R script so that this data can be harvested by the ORKG, you save it as a JSON file, and now you've compiled all of the information that you want to submit to the journal where you want to publish, and you just include the JSON file with your other supplementary data. So it really does not add a lot of time to this process of submitting your paper for consideration. And so this is how we transform scholarly publications into machine-readable literature. And in this case study, we applied this approach to an already published paper. But our vision is to promote a paradigm shift where researchers publish their data in this format from the outset. And to remind you, there are a number of motivations for doing this. One, by creating machine-readable publications, we help researchers navigate the 1,000 new articles published every year on biodiversity loss by allowing new research findings to be automatically harvested by the ORKG. And in this way, we can more easily get an overview of the latest biodiversity trends. And by making biodiversity literature machine-readable from the outset, we can leverage the power of machines to support synthesis research that brings actionable insights to biodiversity conservation. By bringing semantic structure to research contributions with the use of ontologies, we also began to address the challenges associated with inconsistent language and structure across scholarly PDF publications. And by making these scholarly pieces of literature machine readable, we're laying the foundation for more efficient workflows and approaches that could extend the lifespan of syntheses and other publications. And so to wrap up, I want to just briefly mention some of the next steps that we're taking in this direction to address this fourth challenge. So now that we've demonstrated how scholarly literature can be made machine readable, we want to take the next step to apply this approach to conducting secondary analyses. So towards this aim, we just started working on a proof of concept synthesis with the ORKG to demonstrate how this platform can help us to conduct synthesis research more efficiently. And we did this by reaching out to two fellow ecologists, Daniel Paredes and Salma Akhtar, who recently authored papers that are topically related to Ricardo's. And we asked them to share their R code so that we could conduct kind of meta-analysis looking at the effects of landscape composition on pest abundance. And we're really excited that they both quickly responded and shared the code. And we want to acknowledge that they're playing a really key role in helping us to advance this approach. So now we're working to apply the same method that I just showed you with Ricardo's paper to their paper, so that we can make the information that we need for this mini meta-analysis machine readable. This means that it can be automatically harvested by the ORKG. So in this way, the ORKG becomes the database to conduct the synthesis analysis in R. So that means that we as the researchers remain in control of making decisions about how we want to conduct the analysis. But now we have this tool that allows us to efficiently access, organize, and manipulate primary data. And the idea behind this proof of concept is to get a feel for how much we could speed up the process of synthesis research if all of the primary literature were already in a machine-readable format. So we're hoping that this will provide further motivation for supporting this shift to publishing in a machine-readable format from the outset. And this project sits within this broader movement to make scientific knowledge fair. And by fair, we mean information and data that's findable, accessible, interoperable, and reusable. And we're seeing a lot of encouraging steps in this direction. So first, we had these awesome positive responses from our colleagues who were willing to make their code accessible for us to reuse. And this kind of peer-to-peer sharing happens quite a lot. So there's this informal process of sharing information with one another. But in addition, in a more formalized way, many journals in ecology already require us to submit our raw data in a data repository. And there's a growing movement for us to also share our R code when we submit an article for publication. And all of this is in an effort to make our research methodology as transparent, reproducible, and reusable as possible. And our vision is that we can use programming languages like R to facilitate automated data harvesting by the ORKG, and that this will be streamlined into these existing data sharing requirements that are already implemented by many academic publishers. And by ensuring that data from new publications is automatically stored and structured in the ORKG, we make this information accessible and ready for reuse by anybody in any discipline anywhere in the world. So this is just the beginning, and I hope that you stay tuned and you join us to collaborate as we work to transform the way we share biodiversity and other scientific knowledge. So thank you so much for joining the seminar. I hope you found it interesting and thought-provoking, and I'm happy to do my best to address any questions that you might have.